This protocol offers a nanomaterial electrode interface that mimics natural cell membranes, making it possible to determine the effect of localized heat transfer from irradiated gold nanoparticles on the integrity of a biological membrane. The nanoparticle and photothermal effects can be explored in real time on model cell membranes. This technique uses a biomimetic membrane system to observe and interrogate photothermal effects on a biological system, resulting from the heat generated due to the interaction of gold nanoparticles with light, by measuring the conductance and capacitance changes across the model cell membrane. The use of nanoparticles in photothermal therapy is showing great promise as potential cancer therapy. This method provides direct information on how this approach affects cells. This platform provides valuable data to design strategies for thermal therapies that permit optimization of the laser parameters, particle size, particle coatings, and composition. Demonstrating the procedure will be UTS researchers Professor Stella Valenzuela and Dr. Amani Alghalayini, along with Professor Bruce Cornell from Surgical Diagnostics. To begin, carefully take off one coplanar gold electrode slide from its container using tweezers, taking care to not contact the patterned areas where the tBLMs will form. Air dry the slide for 1 to 2 minutes to remove any residual ethanol. Then place the gold electrode over a dry surface with the patterned gold surface facing up. Peel the transparent adhesive layer cover from a thin laminate and place it over the 6 channels to define each well. Use a pressure roller to release any air between the slide and